AgraQuant Gluten G12 test kits are ready-to-use ELISAs that detect gluten in food and environmental samples. In this video, we will cover the contents of the test kit, how to use the test, and how to interpret results. Please keep in mind that this video is not a replacement for the package insert or other important product documentation. To ensure accurate results, please follow the package insert that came with your test kit. AgraQuant Gluten G12 ELISA test kits come complete with ready-to-use extraction buffer and dilution and wash buffer concentrates, antibody-coated microwell strips in a microwell holder, ready-to-use standards, and ready-to-use antibody conjugate, substrate, and stop solutions. Before we start, there are a couple of things worth mentioning. AgraQuant allergen kits require refrigerated storage at 2 to 8 degrees Celsius. Make sure you clean the surface where you will work and the tools you will use. We also recommend that you wear protective gloves and change them before each new stage of the test. Ready? The test works in just a few easy steps. Start by taking the kit out of cold storage. Let it equilibrate to room temperature for about 20 to 30 minutes. During this, you can prepare the extraction and wash buffers and the sample. Calculate the amount of extraction and wash buffer you need according to the number of samples and dilute the buffer concentrates with distilled water. For the extraction, prepare an 80% ethanol solution. Weigh 0.25 grams or 0.25 milliliters of homogenized solid or liquid sample in a 10 milliliter tube and add 2.5 milliliters of extraction buffer, the bottle labeled A. For liquid samples, add only 2.25 milliliters. Incubate for 40 minutes at 50 degrees Celsius and vortex at regular intervals. Allow your samples to cool down and add 7.5 milliliters ethanol 80%. Shake the extract for 60 minutes at room temperature in a laboratory shaker. Centrifuge samples for 10 minutes at 2000 G to obtain a clear supernatant. If there are still particles in the supernatant, you can filter it or repeat the centrifugation. Dilute the supernatant at a ratio of 1 to 10 with the dilution buffer. Your samples are now ready for testing. Remove the necessary amount of antibody-coated microwell strips from the foil pouch and place them in the microwell holder. You can break the strips if needed. Pipette 100 microliters of each standard and the prepared samples into the appropriate wells. Use a fresh pipette tip for each standard or sample and make sure to empty the pipette tip completely. To improve accuracy, you can instead pipette 150 microliters of each standard and prepared samples into transfer wells, and then transfer 100 microliters of each into the antibody-coated wells with a multi-channel pipette. Incubate at room temperature for 20 minutes. Do not attempt to mix the content of the microwells by shaking the plate, as this may cause well-to-well contamination. Empty the content of the microwell strips into a waste container. Fill each microwell with wash buffer and then discard it again. Repeat this step 4 times for a total of 5 washes. After the 5th wash, lay several layers of absorbent paper towels on a flat surface and tap the microwell strips on top of them to remove any residual buffer. Pipette the required amount of enzyme conjugate solution into a reagent reservoir. You need approximately 120 microliters per well. Pipette 100 microliters of enzyme conjugate solution into each microwell with an 8-channel pipette and incubate at room temperature for 20 minutes. Discard the content of the wells and wash 5 times as before with the wash buffer. Pipette the required amount of substrate solution into a reagent reservoir. You need approximately 120 microliters per well. Pipette 100 microliters of the substrate solution into each microwell with an 8-channel pipette and incubate in the dark for 20 minutes at room temperature. Pipette the required amount of stop solution into a reagent reservoir. You need approximately 120 microliters per well. Stop the reaction by pipetting 100 microliters of stop solution into each microwell using an 8-channel pipette. The color will change from blue to yellow. Read the absorbance of the plate at 450 nanometers. This step must be performed within 10 minutes of adding the stop solution. You can analyze the data obtained from the microtiter reader by using the AgraQuant ELISA calculation tool. And that's it. For more information about the AgraQuant Gluten G12 test kit or any of our diagnostic solutions, visit us at rumorlabs.com.